Today we are going to talk about nucleic acids, and the two main ones we are going to talk about are DNA and the RNA. And they're both very similar, they're both involving genetic information. But the difference is that DNA stores the information, while RNA is what codes for amino acids to form proteins. So RNA is kind of the builder, while DNA is more like the blueprint. The thing about DNA is that it can be converted into RNA. The thing about both structures, though, is that they consist of many nucleotides, which we're going to see below. So now we are going to look at something called nucleosides, nucleotides, nucleoside diphosphates, and nucleoside triphosphates. So what are nucleotides? Well, to make up a nucleic acid, we need many nucleotides. So nucleotide is the monomer to the polymer nucleic acid, so DNA and RNA respectively. Now if we look at the nucleoside, we have a 5-membered ring called pentose, and attached it has a nitrogenous base. So what is the nitrogenous base? Well, as I circled here, that is adenine, and it has the N and the H right there, as you can see. And that's for both the A, T, C's, and G's of the other nitrogenous bases, as we will cover next. So what is the nitrogenous bases, the A, T, C's, and G's that I was talking about? So we have DNA nucleotides, so it's adenine, thymine, guanine, and cytosine. But in RNA nucleotides, it's the exact same thing except for the uracil, which instead of thymine, we have uracil. So the thing I do to remember this is that A and G are purines, and C, U, and T are pyrimidines. So the significance of this is that purines and pyrimidines attach, and we're going to talk about this later. So the way I remember this, purines, pure as gold, right? So you have marriage, 2 rings. The importance of that is that purines have 2 rings, while pyrimidines have 1 ring. And the way I remember pyrimidines is pyramid. So top of the pyramid is sharp and it cuts you, and that has 1 ring. So let's take a look at the difference between DNA and RNA. Well, DNA has deoxyribose, so deoxy means it's missing an oxygen. So if we look at the X on this sugar, instead of attaching to an OH, it's attached to just an H. While in ribose, it has an OH. So it is less stable because the OH is more susceptible to hydrolysis. So before we talked about how you can have diphosphates, triphosphates, so how do phosphates attached? So I'm circling 3 things over here, and we basically have 2 sugars and 1 phosphate. So the importance here is that this phosphate is connecting the top pentose to the bottom pentose. Specifically, if we look at the pentoses, there is 1, 2, 3, 4, and 5 labeled carbons. And this is actually important. It's actually labeled like this. So the 3 always has the hydroxide group, while the 5 just has the carbon, right? So when the phosphate attaches both to the 3 hydroxide and the 5 carbon, these bonds are ester bonds. So 2 of them diester. So we have phosphodiester bonds between the phosphates and the sugar. And both of these together create the backbone for nucleic acids, right? So if we zoom in, we look at the DNA, the double-stranded. So in the middle we have our nitrogenous bases, adenine, thymine, guanine, and cytosine. However, the backbone is the white part of the sugar and the phosphate. So now that we know how the phosphate attaches, where does the phosphate even come from? Well, we have the nucleoside triphosphate, which is the whole entire thing we see below. But the thing is, 2 of those, the 2 phosphate groups, cut off. They're pyrophosphate, as you can see on the right. And then all we have left is the nucleotide, so 1 phosphate in the nucleoside, and that attaches to form the chain. So here we go back to the basics. DNA and RNA. RNA is single-stranded and DNA is double-stranded. So the thing is, we have the pyramids and pyrimidines, and the purines are attaching together, as I mentioned before. So we have the adenine and thymine go together respectively, and the guanine and cytosine go together respectively. So we notice that we have the 3' and the 5' and we'll talk about that later. But if you look, the A and the T form 2 hydrogen bonds, while the guanine and the cytosine create 3 hydrogen bonds. So we're going to see this below. So we have the adenine and then at the 3 end and the thymine at the 5 end, and in the middle we have those 2 hydrogen bonds. Recall that to have a hydrogen bond, you need to either bond to HH, HN, O, or F. So those are all hydrogen bonds. And the thing to note here is that the 3' with the adenine and the 5' with the thymine are called anti-parallel because they're opposite, right? But they still connect. So that's called anti-parallel. Here at the bottom, we can also see the hydrogen bonds, the OH and the NH, for the guanine and cytosine, all 3 of them. So that's the end of the video. I hope it was helpful. Please leave a like if you enjoyed the video.